All right, so today what we've got to talk about is some logic problems. Oh boy. So we're going to get into some fairly tricky ground. I don't want you to panic though. We're also going to go over some of this on Wednesday, and I'll have a review after spring break. So we will get through this, even if at first it seems a little bit daunting. All right, so what we're going to talk about today is the triple point cross, but first, let's talk about the lab. So this is just a snapshot of the numbers so far, and they are looking pretty good. So we got almost 1,000 in the F1s, we got over 2,000 in the F2s. There's a few gaps that could be filled in, but I'm not too worried about it. So some of you should have some more things to count this week. I do not, I emphasize this, I do not hold it against you if you can't get large numbers. Sometimes the flies are just really annoying and they don't produce as many progeny as you like. So don't panic over that either. In the end, what we're going to do is we want to just look at this column here. This is what matters. The communal action of the entire class to produce some reasonable numbers. So we'll have to analyze those. And we'll talk about that analysis after spring break. So we'll go through that. This week, what are you going to do with these guys? Well, if any of you have more to count, go ahead and count them. And then everybody gets thrown out. So one of the things I want you to do in lab this week is take all those scarlet and brown flies, those bottles of scarlet and brown flies, and euthanize them and wash out the bottles. And then we'll be done with this experiment, except for figuring out what it means. Then what we're also going to do, we are going to do we've started the mapping cross, the triple point mapping cross. I'm going to be talking about it today. So we've started that, and I'm not really confident we're going to get it done this week. So the idea was, last week and the week before, you started your stocks of white miniature fork flies and wild type flies, and you got them growing. And then what you need to do this week is extract virgin females and cross the virgin white miniature fork females to wild type males and virgin wild type females to white miniature fork males. That's what you got to do. I checked the bottles and they're not really producing a lot just yet. And this is a problem we'll run into again, because this is our situation. You got your wild types, and they're like the Chads and Stacys of the world. And they are going to reproduce robustly. I'll be very surprised if we didn't get our wild types this week. This is the white miniature forked. I'm sorry to say, they're kind of homely with bristles going every which way. They're half blind because they're white-eyed, and they have little short stubby wings, so they can't even sing romantically. So it's a struggle to get them to breed, and we'll see if it comes out. On the plus side, I'm going to argue that these guys are more sensitive and thoughtful and aware of boundary issues. Okay. But just slows them down a little. So that's what we're waiting for, is we want to get the next generation from these guys, as well as these guys, and they're taking their sweet time about it. Okay, if we get to the end of the week and we do not have white miniature fork virgins, don't panic, okay? If you do, that's great. We can get the cross going to be going over the spring break week. If you don't, take those bottles, and I was doing the numbers in my head, and I think they'll still be okay if we get to them promptly after spring break. That should give them plenty of time just to start producing offspring. Otherwise, in lab this week, you might want to repeat this cross and set those up, get them in the incubator, then forget about them over spring break. When you come back, with any luck, we'll have lots of new flies emerging. So it may not work as smoothly as I'd hoped, but we still got time. It'll work. It'll all happen. So we're in good shape. Okay, any questions about the lab? Yeah, it's pretty straightforward. So this week you got a little counting, little bottle washing, hoping we get some virgin females from these flies, and that's kind of it. And then next week you go off and have fun, and then the week after that, we're going to be doing a bunch of counting again. All right, so what are we going to do? Let's talk about triple point crosses. So, last time we talked in general about how you measure map distances in these flies using recombination. So here's a little cartoon illustrating that. So here we got a fly. This is an F1 fly here, so it's a hybrid produced by a cross of a little a, little b, and a big A, big B fly. So we got the heterozygote here. It's going to produce gametes. It can produce 4 kinds of gametes. There's this kind, that's big A, big B, and little a, little b, just like their mom or dad. And it's also going to produce these gametes. These are the recombinant gametes, where there's been a crossover event that now changes around little a and b, so that they're a different orientation from what you saw here. And then we take those gametes and we use them in a test cross. We cross these individuals to a homozygous recessive, and we get a result. So for example, here's some numbers we could get. So in this case, I say, okay, we do this cross and we discover at the end that 45 are big A, big B, 45 are little a, little b, 5 are big A, little b, and 5 are little a, big B. These are the recombinant types. The only way you'll get these is if there is a crossover event between those two loci. Okay, so that's what we need. And then you can use that information to calculate the recombination frequency, which would, in this case, would be 5 plus 5, these recombinants, over the total. So 100 total here. So recombination frequency is 0.1 or 10%. And then we would say, the map distance between A and B is 10 map units. Okay, so you all got that last time, right? No problem at all. There's a term I will use occasionally on quizzes and homework and things like this, and I just want to bring this to your attention so you know what I'm talking about when I say it, and that is, what is the arrangement of the alleles? So what I'm doing there is I'm asking for the arrangement of the alleles. In this case, go back. In this case, we're looking at the A's and the B's. When I say, what is the arrangement of the alleles in the parent generation? And these guys right here, you would be able to say, okay, on one chromosome we got big A, big B. On the other chromosome we got little a, little b. That's what I mean by arrangement of alleles. There's another possibility. What if it was this? Big A, little b, or little a, big B. It makes a huge difference in your calculations. But it's also really easy to determine what the arrangement of the alleles are. All you need to do is look at the non-recombinants, and that tells you. Okay, so here's an example. Here what we've got is, did I go the wrong way? Oh no, I did. It's okay. So here's our first example where I said, okay, we got this arrangement of alleles here. On one chromosome set, we got big A, big B. On the other one, we got little a, little b. And we calculated the distances there. Here's another possibility. What if I give you a problem and I say, okay, these are the results of our test cross. We're going to have to figure this out in reverse. And we're going to ask, what is the arrangement of the alleles? And you would look at this, no further thinking required, right? You just say, oh, well, the arrangement of the alleles have to be big A, little b, and little a, big B in the F1 parent. Everyone sees that? It's as simple as just looking at the non-recombinants, and it tells you so much. And if we think backwards, okay, then still the recombination frequency is 0.1 or 10 map units. Nothing has changed there. We're going to do a test cross. Okay, there's our various chromosomes. So here's the non-recombinants, and you can see big A, little b, little a, big B. And here's the one where we had a crossover event. So in this case, this arrangement is the recombinant type. And that tells us that the parents had to look like this. So you can see those two I've just shown you are exactly the same problem, just with a different arrangement of alleles in the parents. That's the only difference we made. Okay. Now, what we ultimately want to do with this is some more elaborate mapping experiments. I mentioned this one last time. This is Alfred Sturtevant did this really basically an observation that Thomas Hunt Morgan had a collection of these pairwise distances. So we just measured AB is 10. There's a catalog of those for all of these different pairs of genes. And Sturtevant took those together and he said, you know, I can assemble these. So here, for instance, it says yellow and white are really close to each other. They're about 0.5 map units here. And white miniature are a little farther. And there's yellow. It's farther still from miniature. And he looked at that and he saw there's only one logical way that you can fit these together so that the numbers all add up. So this is a common approach to mapping. You get two at a time, and you get another two, and you try to find a link between them, and you sort of assemble them into a completed map. So that's what Sturtevant did. Now, another kind of problem I could give you sometime is like this. I would say, okay, here's a little problem. Say, okay, we got A to B is 10 map units. And I also do another experiment, and I figure, in the same way that A to C is 5 map units, and B to D is 3 map units, and C to D is 18 map units. So I've worked I've done all these crosses, I've made all these measurements. I got these 4 distances, and you have to put them together into a map. So how do you do that? Well, what you can do is we can just start with 1. It doesn't matter which one, actually. So there, we'll start with the distance from A to B is 10. There's my chromosome. I know that much about it. Then I say, okay, we got A to C over here. Look at there. There's A, and the distance to C is 5 map units. So let's put it there. Do you see a problem? Is that the only place C could go? Where else could it go?
Between A and B.
Yeah, it could go the other way. So we have to keep that in mind. So we might sketch out a little map like this, where tentatively we say, okay, C is either over here or it's over here. We're not sure yet. We got to find some connections, and then we'll puzzle it out. And then we say, okay, B to D is 3 map units. So yeah, it could be there. And just like the previous one, maybe it's not. Maybe it's over here. All we know is it's 3 map units either way. But you see, now we got these compounding, multiplying possibilities for the map. But then fortunately we got this one last measurement, C to D. C to D is 18 map units. So there it is. So does that tell you where C and D have to be? Yeah. So now we know, okay, we can get rid of that one, we can get rid of that one. It must be C, A, B, D is the order of the genes. And we can make a map that kind of looks like that. So that last measurement kind of helps us lock in where the positions are. What if we'd done this and I said, okay, the distance from C to D is 2 map units? Would you do a different map? Yeah, then you'd say, okay, well then C must be here and D must be here. They add up nicely, they're 2 apart and all that kind of good stuff. So that must be the map. Notice that, as we'll discuss in a moment, measuring map distances this way, it's not like pulling out a ruler. You're looking at statistical variation. So sometimes it won't be exactly spot on. Again, if you think back to Sturtevant's results, they didn't exactly add up either. But you're just trying to get close enough to reach the most logical arrangement of the genes in the cross. Okay. I will have some more problems like this for you to do in class on Wednesday. Okay, here's the other thing. So I showed you this. I said, okay, the frequency of recombinants take the number of recombinants divided by the total number, that's the frequency of recombinants, is the same as the map distance. Is it always? There's a problem looming here. Because there's another possibility. These aren't the only kinds of gametes that can be produced. What if there are 2 crossovers and they both happen between A and B? So we get 2 crossovers. So the first one flips the order of the A and B alleles, but then the second one flips them right back. That means you won't see it. It'll be totally invisible. So these are invisible crossovers. And what we wanted to say is, okay, the recombination frequency is the same as the map distance. But it's not, because we're constantly underestimating the number of crossover events between any 2 genes. Because those pesky double crossovers, they hide from us, so we don't see them. So what are we going to do there? Okay, so like it says there, double crossovers. They are crossovers, but when we do this experiment, they're counted as non-recombinants. So we're going to be off a little bit. This has been plotted out. So here, for instance, this is the theoretical map distance. That if we measure percent recombination, and like I said, if we measured 40% recombination, that would correspond to 40% map distance. That's the ideal. When you actually do the experiment and start measuring things, what you find though, is that the observed recombination frequency is always lower than you expected. And that's because those double crossovers and quadruple crossovers, any even number of crossovers between 2 loci, is going to mean you don't see them, they're invisible, so you're going to miss them. in this kind of analysis. Now, this does show, though, that at least down here, when you get really short map distances, if you get really short ones, it's unlikely you'll have two crossovers in the same place. So down here, it's pretty accurate. So if you're looking at two genes and they're like 5 map units apart, you know, what you'll see is pretty good. It'll be a pretty good measure just looking at the recombination frequency. If they are 30 map units apart, ooh, you're going to be off, significantly off. Now, just to warn you ahead of time, the triple point cross we're going to be doing in the lab, the white miniature forked, those genes are spread out over, I think it's like 40 or 50 map units. So we're going to see some inaccuracies. You'll have to take that into account when you write up the discussion for your lab report, but right now don't worry too much about it. It just means we're not going to see perfectly spot-on numbers to what they should be, and it's kind of okay. The reason I picked 3 genes that are so far apart is that we want to see a reasonable frequency of getting double crossovers. And you guys don't want to count 100,000 flies, right? No, you're supposed to say, yes, you're enthusiastic. We'd love to count 100,000 flies. Yeah, you just have to stay over Christmas break. No problem, right? No problem. Okay, so we've got a little trade-off there in the experiment we're doing. In the real world, in a real genetics lab, they would not do genes that far apart. They do a series of them, closer together, and then like in the previous method, we'd sort of stack them up and get a more accurate measure of the distance. Okay, so here's our solution to that problem. That double crossovers make everything inaccurate. And that is, what if we threw a gene in the middle? So here we got ABC. We want to know the distance between A and C. Sure, just throw in another gene. Throw in a B here to fill in that gap. And then we can measure not only the distance between A and B and C, but we can also measure how often double crossovers occur, which might be useful information. So that's going to be our solution. That's why we do a triple point cross, is because it will give us additional information about the frequency of double crossovers. This obviously suggests, why don't we do 5 genes at once? Yeah, put another gene in here in this gap so that we can detect triple crossovers and quadruple crossovers and all these little things. Put another one there. Yeah, it gets kind of unwieldy, doesn't it? 3 is enough for now. We'll work with 3. Okay, so this again is a method called the triple point map. And here's the basic procedure. This is what you're doing in the lab, starting right now. Is you begin with an individual known to be heterozygous for 3 linked genes. So I gave you white miniature fork flies that are homozygous for 3 genes. And I gave you wild type flies, which are homozygous for the wild type alleles for those genes. And what we're trying to do this week, and as I said, I'm not sure we'll make it, but we'll try. So what we're trying to do this week is get those together to breed and produce heterozygous flies that are heterozygous for white miniature and fork. So that's our goal. Then we take that individual and we cross it to a homozygous recessive individual. We're going to make a test cross. That will be after you get back from spring break. Once again, I have designed this experiment so that you don't need to worry about virgins with the next cross. We're going to just breed brothers and sisters, and because the way it's set up, you'll see, it will work it all out. And then what you have to do is look at all the progeny of this cross. You want to count ones that are non-crossovers, single crossovers. There'll be two kinds of single crossover flies, and double crossovers. So yeah, this cross you just finished, you just had to score for wild type, red eyes, scarlet eyes, or brown eyes, right? And albino eyes. So we got 4 traits you were looking at there. What you're going to be doing in this next cross at the end of the semester is you're going to be looking at 8 traits. 8 different combinations. So see, we're just kind of ramping up the difficulty a little bit on you. But you're pros now, you'll have no problem. Okay, so this is what we want to do with our 3-point map. And the key thing is including that third gene allows us to detect double crossovers. Okay, so what would they look like? Here's just an arbitrary example. Here we go again. Big A, big B, big C. With little a, little b, little c. And we're going to look at the gametes it produces. And what it's going to produce is, okay, there's a non-crossover gamete right there, right? That would be big A, big B, big C. Similarly, this is a non-crossover here, little a, little b, little c. There they are. If there is a double crossover here and a crossover here, the double crossover gametes will look like big A, little b, big C. Right? And you can also see little a, big B, little c. So those would be the double crossovers. We already know what to expect. We're going to be looking for those. Not shown here are the single crossovers. What would the single crossover gametes look like? Again, you can imagine, okay, big A, there's a crossover, little b, then we don't have that crossover, that would be then little c. So big A, little b, little c, or its complement, little a, big B, big C. Alternatively, if this crossover doesn't occur, it would be big A, big B, little c, and little a, little b, big C. You get the logic? That's what we're looking for. So we're only showing the double crossovers and the non-crossovers here, but there's going to be two classes of 2 types each for single crossovers. See, it's already getting messier, isn't it? Okay, so what I'm going to do now is in your textbook there is a figure. It's figure 5.8 in my book. I don't know if they renumbered it. You'll see it in a moment. You'll recognize it. But yeah, you all have this diagram in your book too. And it's doing the same thing. So we're going to start with a heterozygous individual. The genes in the diagram are yellow, white, and a kinase. So this is yellow body, white eyes, and rough eyes. And we're going to cross that in a test cross to an individual that's homozygous recessive. In this case, we're using the fact that these are sex-linked. So we're going to cross it to a male that's hemizygous. That means we're actually going to cross the brothers and sisters. Again, we'll go through this specific experiments in detail. And then we're just going to count all the progeny. Easy, right? You know how to count flies. So what will it look like? Here's that diagram. I'm going to go through each piece in a little more detail, so I'll zoom in on some of these things. But here's our cross. And for our purposes, what we're going to care about is just this stuff over here. Okay? We're going to look at the phenotype of the F2s. So we've done a test cross, we produced the F2s, and we're going to ask how many of each phenotype is there going to be. And as you can see, there's 1, 2, 3, 4, 5, 6, 7, 8 categories. And if you start counting sex, that means there'll be 16 categories. Yeah, it'll be loads of fun. And then we have the numbers that we got over here. And the end result is we're going to get a map that looks like this. But I'll show you how we get to that point. Okay, so let's first we'll start with the easy one. Let's look at the non-crossovers. The ones where there was no recombination. How do you identify those? You just look over here. So there's a total of 10,000 flies in this experiment. And here you can see 4,685, 4,759. So we got a total of 9,444. The non-crossovers will always be the most numerous. So we can immediately pluck out which of these categories is the non-crossover category. And then we can look at their phenotypes, and there they are. Yellow, white, and echinus versus wild type yellow, wild type white, and wild type echinus. When you pull out these groups and lump them into the category, in this case the non-crossover category, you're going to do that first on the basis of the number, but then you should always double check, do these complement each other? And they do. So this one's yellow, this one's wild type yellow. This one's white, this is wild type for white. This one's echinus, and this one's wild type for echinus. So you do that, just sort them on the basis of numbers, then make sure that the categories are complementing each other. Okay, that's the non-crossovers. What about the single crossover events? That is a crossover like right there or right there. What will we get? So here are the single crossover categories. The single crossover categories will be the second most numerous. Hey, this is easy, right? You just gotta look at the numbers. You can tell which one goes in which category. In this case, what we've got is 80 and 70 for this group. And again, you look and see, do they complement each other? Yeah, this one's mutant yellow, but wild type yellow. Wild type for eye color. This one's mutant for eye color. This one is wild type for echinus and mutant for echinus here. So we can satisfactorily lump those together into this one category of the single crossover. Furthermore, since this is the arrangement of the alleles, this is the order of the this particular example makes it easy because they tell you here's the order. White's in the middle. They won't always. We'll see how to figure that out later. So we got this order, and so to get this arrangement, there had to have been a single crossover between yellow and white. Okay? So we got that, and here it says, okay, that's 1.5%. We got 150 total, that's 1.5% of the total have a crossover between yellow and white. And you might want to say, oh, well, that means the map distance is 1.5. Not so fast. We've got a little bit more to talk about. And down here we see this other category. 193 and 207, total of 400. And we got, again, they complement each other. Always double check that. I can think of ways to devise problems where they don't complement each other. So we'll get to those eventually too. Okay, so they complement each other. When we look at this order of the alleles, we can say, well, there must have been a crossover between white and echinus like this. So this is telling us roughly the distance between white and echinus, just like this one is telling us the distance between yellow and white. All right, now the fun part. Double crossovers. Double crossovers will be the rarest of the bunch. So notice here, we only got 3. 3 here and 3 here. We double checked. Yes, they complement each other. This is the complement of that. There's a total of 6 So our frequency is 0.06%. So this is a rare event. And the only way you can get yellow with wild type eye color and a kindness is if there were two crossovers like that. All right, now for the tricky part. So I told you previously, okay, we saw a map distance of 1.5 between yellow and white, and 4.0 between white and echinus. You now have to add this frequency to both of those numbers. And every year, this kind of blows some students' minds. You got to add that frequency twice? Yes, you have to add the frequency twice. Because these double crossovers represent a crossover between yellow and white and a crossover between white and echinus. So we have to add that 0.06 to the distance we measured for yellow and white, and also to the distance between white and echinus, in order to get a more accurate map. Okay. Any questions about this? I'll give more examples. We'll go over some more. Don't fall asleep on me. Got to think these through. But basically, these triple point crosses, these 3-point mapping, this is all a logic game. Got to work through the logic of each step to figure this out. All right, so what's next? Oh yeah, that diagram cheated because it gave you the order. It told you what order those loci are on the chromosome. It just flat out told you white's right there in the middle between yellow and echinus. That won't always be the case. Sometimes you won't know before you start. So one question I might ask is, how do we know that the order isn't this? How do we know that yellow isn't in the middle or echinus? Other than the fact that the textbook told us so. So sometimes we have to figure out the order as well as these frequencies. So here's possible results. If yellow were in the middle, then the double crossover sequence would be white, wild type Y, echinus on one of the chromosomes. And that wasn't the case. That doesn't work. That's a big distance. So when we look at the number of categories, or number of flies in each category, this one wouldn't fit a double crossover. The double crossover has to be the rarest of all the possibilities. Similarly, echinus can't be in the middle, because then this would be the double crossover phenotypes. These two. And again, those were too numerous. So it has to be this one. And I'll tell you how to instantly figure out which one had to be in the middle in a moment. Okay, let's look at another problem. This one's trickier because I'm not telling you anything. Okay, we got brown midrib, we got virescent seedling, and we got purple aluron. This is all on a plant in this case. And we start with a female plant that's heterozygous for all three, and a male plant that's homozygous for all three. But we don't tell you these crucial things. We don't tell you the arrangement of the alleles of the female. Remember the previous example? We made it simple and obvious where all the wild type alleles were on one chromosome and all the mutants were on another. That's not necessarily true. You've got to figure that out. We aren't going to tell you which one's in the middle. That's another secret that will be revealed by the results of the cross. And obviously we haven't told you the distance between the genes. So in this example, we're going to have you make another map. And the only information you have, the textbook isn't telling you anything other than the frequencies of the different results. of the different phenotypes that result. Okay, once again, we got to think about which one is in the middle. That's when we ask what's the order of the genes. That's the simple version of the question, is which one is in the middle? And as you can see, there's multiple possibilities here. So here, PR could be in the middle. Here, BM could be in the middle. Down here, V could be in the middle. How confusing. How do we sort these out? So we don't know which one is correct yet until we do the cross. So here's the result of the cross. Okay. So we cross those and we get these results. All right. There's the most numerous category. So what you would say right away is, okay, most numerous one, that's got to be non-crossover. You all agree right there, right? Yeah, okay. So we look at that and we say, okay, these have got to be the non-crossover phenotypes right there. And we look at those, and do they complement each other? Yes, they do. So that's all good. So there's our non-crossovers. And then what you want to do is look at the double crossovers. Once again, the double crossovers are the rarest. The rarest phenotypes are going to be these guys. Look at it, there's 86 of those. Much rarer than either of these others. So this has to be our double crossover. And then we look at the phenotypes of the double crossovers. The first thing we do is we check, do they complement? Yes, they complement. So we're doing good so far. We can trust this result. Now we want to know which one is the middle locus. And here's the easy way to figure this out. You want to just compare a double crossover phenotype to one of the non-crossover phenotypes. Okay, so PR, V, BM, and this one is plus V, BM. Which one is in the middle? It's always the odd man out. The one that's different. So say PR, V, BM, and this one's plus V, BM. Which one's different? PR. So that tells us PR has to be the one in the middle. Now when you're solving this kind of problem, one of the things I suggest is you quickly figure out which one's in the middle and then rewrite the order of the genes. So yes, the genes are all listed here. You've got PR on the left. I find that hard to visualize, but if I write it out this way, so just now PR is in the middle, we just switch those around. And this is the order of the loci and the arrangement of the alleles. So this is the arrangement of the alleles in the parents, and PR is in the middle. Now, why would I do that? Why would I write it out this way? Because it makes my life so much easier. Because here, for instance, here's a single crossover event. All right, we see we've got 14.5% in this category. And yeah, they complement each other too. But where did the crossover occur? It's a single crossover between which two loci? And I find if I rewrite it this way, I can see how can we convert this into this? How do we get a non-crossover to look like that? There had to have been a crossover event between V and the middle one, PR. Right? So you see that right there. So that's telling us this is a single crossover between V and PR. And if I was solving this on a little piece of paper, I'd actually make a note. I'd say, okay, single crossover between V and PR, right over there. And this one, then when we look at it, how do we get this from this non-crossover? There had to have been a crossover between this one, BM, and PR. So that's a single crossover event here. And then obviously by definition, a double crossover event like that one is between both of them. Okay, we're almost done figuring this problem out. So we make a little note over here and say, okay, this is the single crossover between V and PR, and this is the single crossover between PR and BM. So when I look at this frequency, this is the start of figuring out the distance between V and PR. And this frequency is going to help me figure out the distance between PR and BM. Is anybody lost? You all seeing how to do this? Again, we'll go through these step by step a couple times to make sure everyone grasps the concept. All right, but if I want to know the distance between PR and BM, for instance, it's got 35.6 here, but we also have to add that 7.8 onto it, because that is also a crossover between PR and BM. So the distance between PR and BM is going to be 35.6 plus 7.8, which is 43.4. Likewise, distance between V and PR is 14.5 plus 7.8, which is going to be 22.3. All right, and here's the final calculation. We can just do all this stuff. There's our map down there at the bottom. It's got a couple of features to it, right? We now know the order of the genes with PR in the middle. We know the distance between those. This is just going through the different possible arrangements to figure out which one is the double crossover, but I just told you that's easy to do. Just compare the double crossover category phenotype to the non-crossover phenotype, and one of the loci will be different. And that's the one that you know is the middle one. Okay. Are you ready for a third example? Yeah, we got to go through another example because trust me, on Wednesday I'm going to hand you these problems and I'm not going to help you. You're going to have to figure it all by yourself. So let's go through the steps one more time. So in this case, we've got kind of an abstract example. We've got A, B, and C. We start with a parent heterozygous for those. We do a test cross. Does that sound familiar? Should be by now. And we get these phenotypes. And this is the number. And I'm asking you, what's the arrangement of alleles in the parent? What's the order of the genes? Which one is in the middle? And what's the distance between them? So let's start with the first question. What's the arrangement of alleles in the parent? How do you figure that out? What are you going to look for? Yes?
Those are the highest observed numbers?
Yeah, it's that easy. Look at there. 3980 and 4018. Those are distinctly much more numerous. So we know that the non-crossovers, the parents, have to be this one and this one. And when you compare those, hooray, they do complement. So that works. So what's the arrangement of alleles in the parent? Again, all you gotta do is look at those. You just gotta look at those non-crossovers. So in the parent, on one chromosome, it had to have been big A, big B, little c. And on the other chromosome, it had to be little a, little b, big C. Okay? But now I want to know which one's in the middle. Which of those three genes is in the middle of the sequence? And you've done the first step. We've got our non-crossovers. Maybe I should write those down over here. Let's do, oh, give me a pen. Okay, so we just said, okay, the non-crossovers are big A, big B, little c. And its complement, which is little a, little b, big C. Okay, so this is non-crossovers. What are the double crossovers?
Big ABC and little ABC.
Yes, again, it doesn't matter if they're homozygous, all that matters is their numbers. But yes, so we got one category that's 42 and another one's 45. Do they complement each other? And yes, they do. So our double crossovers are going to be big A, big B, big C, and little a, little b, little c. So we figured out the non-crossovers, and we figured out the double crossovers. Which gene is in the middle? C. How did you get that?
Because if it was any other, it would be either big A, small b, big C, or some other arrangement. But because it turned into a homozygous.
Yeah. Yeah, so we can, for instance, just compare those two. And the different one between those two is C. So that tells us, okay, C is in the middle. We could also do something like, oh, what if we compared these two? It really doesn't matter which two from the category you compare. You compare those and you see these two are different, that one's the same. So it's the C again that stands out. So that tells us C is the answer. What if we compared that one to that one? And C is the weird one. So everything in this result is telling us C has to be in the middle. Okay, so like I said, if I were solving this, I would immediately rewrite the order. So it'd be A, C, B in every case. It'll make it easier to figure out what's going on. Okay. So here we go again. This is just the same thing. Most numerous class is the non-crossovers. There they are. And the parent had to be big A, big B, little c, little a, little b, big C. As it says, this does not mean the order is ABC yet. We're going to do the next step, which was compare the double crossovers. So we look at the least numerous category. There it is. Again, we double check the complement, so it must be right. There's the numbers, and when we compare that to the non-crossovers, C is the weirdo. So C jumps out as the different one, so it's in the middle. Then we have to figure out the distances between these genes. Now, I didn't do it here. I should have done this. I should have rewritten these to put C in the middle, right? Then it would be clearer. But the first row, this group here, is telling us the distance between A and C is 12.02%. Whereas this one is the crossover between B and C. That's 7.13%. Double crossover is 0.87%. And then we have to add 0.87 to that, 0.87 to that. And we get the final answer, which is the distance between A and C is 12.89%. Distance between B and C is 8%. See, these are not scary, are they? I always get students who just get lost somewhere in this discussion. So again, Wednesday we'll have more examples and make sure you all know this. All right. Now, sometimes what happens is you do this comparison, and like I told you earlier, you do the comparison and the numbers don't add up perfectly. So in this case, the distance from A and C to B, if we add those up, 7.13 plus, add this one up, 12.89 and 8.0%, it says that the distance between A and B, remember C is in the middle, should be about 20.89%, almost 21%. Let's see if we step back. Oh, I didn't do the calculation. Darn. But if you do the calculation and just ask, okay, what's the frequency of the non-crossover types, it's going to be different. So what you have to do is trust the distances between the individual genes and add those up, because they're shorter distances, so they'll be more reliable. Oh, do I want to talk about this? Ooh. How are your brains doing so far? Pretty good? It turns out there's other complications. So like we've been talking about two-strand crossovers, so a two-strand double exchange like this one. In the real world, when you actually look at the chromosomes, what they're doing, you can also have three-strand double exchanges, like this one. So this one's between this strand and this strand, this one's between that same strand there, and the second strand. In that case, even if you've got a double crossover between two genes, it's detectable in the frequencies of the progeny. And here, Nightmare City, if you've got four-strand exchanges like this is kind of like two single crossover events. They're involving completely different chromosomes. And those are all detectable. So when we actually look at the results of a cross, we're kind of balancing all of these to try and figure out what's going on. But these, again, are invisible to us. We don't have a way, using the techniques we're going to use in this lab, to discriminate between 2, 3, and 4-strand double exchanges. One last number I'm going to throw at you. So remember I said one of the reasons we want to do this is we want to know the frequency of double crossover events. How often do we get double crossovers? And this is what we're going to look at here. So, for example, in that PRV and BM example, we figured out the distances between PR and V was 22.3, and the distance between PR and BM was 43.4. So then we might ask, how many double crossovers should we see? Again, remember, these distances are correlated with the recombination frequency. So we're saying there's a recombination event between PR and V, and there's a recombination event between PR and BM. What's the likelihood of getting both in the same time? To put it another way, what's the probability of getting a crossover between V and PR and a crossover between PR and BM? Remember way back at the beginning of the course we talked about probability problems? And when I said, and, what do you do? Yeah, I know, that was 8 weeks ago. Can't possibly remember that. But yeah, if I say, what's the probability of event A and event B happening at the same time? You multiply the probability of the individual events. So one thing we would say is, we should see double crossovers with these distances by taking 0.223 and multiplying it by 0.434. And that will give us the probability of both crossovers happening. And if we do that, we can say, we ought to see double crossovers 9.6% of the time. So it's not that uncommon an event. But when we do the measurements, if we go back to that data, you'd see that the actual number of double crossovers was 7.8%, not 9.6%. So we could ask this question: what percentage of the expected crossovers were actually observed? And there we have to do something a little different. We have to divide. Yeah, you can do that. So this is observed over expected. So we got 80% of the crossovers that we expected to see. It was a little lower than we expected. That gives us this, allows us to figure out this little number here. It's the coefficient of interference. Okay, this might have something to do with a mechanical process. Okay, you got these chromosomes, you're having a crossover, it's forming chiasmata at one point. Maybe the chiasm interferes, it gets in the way of making a second crossover too close. Yeah, if you think about it, you got two little X's there. If they're stacked right on top of each other, that's really hard to manage physically. So we can calculate the coefficient of interference, which is 1 minus observed double crossovers over expected double crossovers. Okay, that's what we just calculated there. That's observed divided by expected, 0.8. Coefficient of interference is 1 minus that. So in this case, our coefficient of interference is 0.2. All right, again, if there was complete interference, if nothing was allowed to have a second crossover event, you would have an I of 1.0. That's the maximum. If, on the other hand, every time you had a crossover, there was another crossover right next to it, it was always making double crossovers, then you would have an I of 0. So the coefficient of interference is going to vary between 1, or between 0 and 1.0, where 1 means that there's a colossal amount of interference. There are some organisms where we see this. In nematodes, for instance, nematodes only allow one crossover per pair of chromosomes. So yeah, if you've got one crossover in this chromosome, it's just going to say, nope, sorry, no more crossovers allowed. So in those cases, it would have an I of 1.0. Okay, one last number I'm going to throw at you. And this is the coefficient of coincidence. Okay, coefficient of interference tells us how much a crossover event interferes with another crossover event near us. So high I means crossovers suppress other crossovers in the neighborhood. Coefficient of coincidence is just kind of 1 minus that number. So if you've got this one would say if you've got a high C value, it means it promotes more crossovers in a particular region. You get one crossover here, and it's like the molecules involved in generating the crossover event are going to appear here and facilitate another one in the same place. So that's just the observed DCO over the expected DCO. You remember what that was in the last example? It was 0.8. It's also 1 minus I right there. And I was 0.2, so it's 0.8 again. So again, it's just a useful number. Depending on your perspective, do crossovers suppress neighboring crossovers, or they promote additional crossovers? Okay, that's a good place to stop. So Wednesday, I will come in armed with problems, and we'll work through them. And then you're going to go away and have a great spring break, right? Yes, all right.